Welcome back to Unit 6 of Bio 150. With this unit, we're going to talk about controlling microbial growth. So we've got to get into talking about some basic terms first when we talk about controlling this microbial growth. So with this, some of these terms, one important one that you see down there is sterilization. That's towards the bottom. That's simply going to be the removal or destruction of all microbes in or on an object. Next, we have what's referred to as aseptic. That's environment or procedure that is going to be free of contamination by pathogens or of pathogens. Disinfection, that's going to be use of physical or let's say chemical agents known as disinfectants to inhibit or destroy microorganisms. And so when you look at your disinfectants here, that might be some things like phenolics, alcohols, aldehydes, soaps, things like that. And we're going to discuss those later on in the lecture. You have disinfectants. These are going to be the use of physical or chemical agents known as disinfectants to inhibit and destroy microorganisms. So with disinfection or disinfectants, that's actually going to be like the terms or the brands that you're going to use for those. Next, we have antisepsis. Antisepsis is going to be when a chemical is going to be used on the skin or other tissue. Now, these chemicals are going to be different from aseptic, things like that, because these have to be safe for the tissues and not destroy the tissues, but still eliminate and reduce the number of microorganisms. Degerming is going to be the removal of microbes from a safe surface or by scrubbing it. So with this one, you're going to go in, you're going to remove these microorganisms from that surface by taking usually some type of, you could use a disinfectant, you could use something that's aseptic, or you can just simply scrub really hard to remove those microorganisms from that area. Sanitization is going to be the process of disinfecting to reduce the number of pathogenic microbes to meet an acceptable public health standard. Pasteurization, we talked about this with Louis Pasteur. This is going to be the use of heat to kill pathogens, and it's going to reduce the number of spoilage microorganisms that are going to be found in food and beverages. And then some suffixes that you need to know. You can have stasis or static. That means to inhibit the microbial growth. You can have sides or sidal, and that's going to be to destroy or permanently inactivate microbial cells. So with microbial death rates, the microbial death is going to be the permanent loss of reproductive ability under ideal conditions. So it's no longer going to be able to reproduce. Microbial death rate is going to be used to evaluate efficacy of antimicrobial agents. So determine if they are useful or worth what you put into it. So usually these are going to be found to be constant over time for any particular microorganism under a particular set of conditions. So constant death rate is going to equal a straight line. And so that's what you should see there going downwards. Antimicrobial agents, you're going to have two modes of action for these antimicrobial agents. You can have what's known as a cell wall and membrane alteration. So what happens here is going to affect the ability of that cell wall to prevent the cell lysis as water moves into it by osmosis. And so lysis is going to be when it bursts or becomes so full that it pops. And so it does that one. And membrane damage can cause cellular contents to leak out, causing that cell's death. Protein and nucleic acid damage. So you could have damage to the proteins that affect the ability of that protein to function correctly, which would cause death. And you can damage the nucleic acids, and that can cause a fatal mutation and a stop in protein synthesis that cell needs. So it can't make the proteins it needs. Factors that are affecting the efficacy of antimicrobial methods, it could be the site to be treated. So with that method and level of microbial control used is going to be dependent upon the site that you need to treat. So harsh chemicals and extreme heat cannot be used on tissues that belong to like humans or animals, anything that's living, because you don't want to destroy the tissue, you just want to remove those microbes. It's also going to be relative to susceptibility of that microorganism. So the method and level of microbial control is going to be used to select and kill the hardest microorganisms first, along with more fragile microbes as well. You can use things like germicides. Those are your antimicrobial agents that are used to kill certain pathogens, microbes, and destroy their structures. And these are going to be classified in 3 ways. You can have what's known as a high. That's going to kill all pathogens, including the endospores. Remember, the endospores are going to be how what they go into, kind of like a dormant phase when they're not active. You can have an intermediate, and that's going to kill fungal spores, protozoan cysts, viruses, and pathogenic bacteria. And then you can have a low. That's going to kill the vegetative bacteria, that means the ones that are actively growing, fungi, protozoa, and some viruses. Now we're going to talk about selection of microbial control methods. We're going to have different factors that affect the efficacy. And with this one going in, we're going to have those environmental conditions. So this is going to be a method and a level of microbial control that can be used in conjunction with ideal temperature and pH conditions, and that makes it prove more effective. So some methods are going to work better with higher temperatures versus cool temperatures, and some methods work better with lower pHs instead of higher pHs. So they work better, let's say, for example, higher temperature and more acidic or lower pH. Now going into the antiseptics here, disinfectants and antiseptics, we have what's called the phenol coefficient. This is going to be a ratio that compares a given agent's ability to control microbes to phenol. So greater than 1.0 is going to be more effective than phenol. If you have a larger ratio, it's going to have a greater effectiveness. You can use what's called a dilution test. That's going to be the current standard test that we're going to use in the United States right now. That's going to take metal cylinders that are dipped in broth cultures of bacteria. They're dried, and then they're going to be immersed into different disinfectant solutions. After 10 minutes, the cylinders are then going to be incubated. Most effective agent is going to be one that entirely prevents microbial growth at high dilution rates. You could also have what's called the Kelsey-Sykes capacity test. This is an alternative to the use of the dilution test. It's standard in European Union, so it's standard across Europe. Chemicals here are going to be added to the broth cultures of bacteria, and samples are going to be incubated for about 48 hours. Then they're going to look at the turbidity, and that's going to be used to determine the minimum time required for effectiveness of agents. In-use test is a 4th test that we can discuss. That's going to be a more realistic method. Basically what they do here is they're going to take swabs from an actual object before and after the agent application. So swabs are then going to be inoculated into a growth media, and then they're going to be examined after the incubation. This is going to provide a more accurate determination if that agent was effective. So it's going to give a better efficacy if it worked or not. So some physical methods of microbial control. We have what's called the heat-related method. So high temperatures are known to denature proteins. Remember, that is go in and you break the bonds which change the shape. If you change the shape, it's going to change the function, so they won't function properly anymore. This can also interfere with the integrity of the cytoplasmic membrane and cell walls of that cell, and it can disrupt the function and structure of nucleic acids, so it messes up the DNA. Thermal death point is going to be the lowest temperature that's going to kill all cells in the broth culture within 10 minutes, so that's going to be important. That's the thermal death point. You also have thermal death time. That's the time that it takes to completely sterilize a particular volume of liquid at a set temperature. Decimal reduction time is going to be the time required to destroy about 90% of the microbes in the sample. So decimal reductions, 90% of the microbes. Thermal death time is 100%. So you're going to have 2 different types of heat-related methods. Now we discussed the heat-related death. First, you can have what's called moist heat. The most effective type of heat is that moist heat. So it's going to kill cells by denaturing the proteins. So we're breaking their shape, breaking their bonds, and destroying their cytoplasmic membrane. So 4 different moist heat methods. First one's going to be boiling. That's effective in killing vegetative cells of bacteria and fungi, protozoans, and most viruses. It doesn't effectively kill the endospores, though, or protozoans that are in cyst form, and it doesn't kill some viruses. Tendilization is going to involve subjecting liquids to alternative periods of boiling and cooling over extended periods of 3 days. It's effective because it maintains liquids at room temperature. Typically, that's going to cause the endospores to start germinating or start growing again. And the result of these vegetative cells now, that they can be killed and denatured in the next phase of boiling. So you might kill everything that's there, and if the endospores live, if you allow it to come back to room temperature, they might germinate, which means they go into an active growing phase and are no longer an endospore phase that can protect them. And that's going to allow it to actually go in and kill these new germinating cells. So it eventually eliminates all of the dangerous microbes. We have autoclaving. That's going to be a process of applying pressure to boiling water, and that increases the temperature of the water. So the conditions required to destroy all microbes is going to be at 121 degrees Celsius, that's 250 degrees Fahrenheit, and about 15 psi of pressure for 10 minutes. So basically, an autoclave is kind of like a pressure cooker that you would put food in as you were canning or cooking. We have pasteurization. That's going to be a method of heating food to kill pathogens and to control spoilage of organisms without altering the quality of that food. Not considered sterilization because heat-tolerant microbes do survive in this type. We'll have 3 methods. You have what's called the historical batch method, and that's where you get to 63 degrees Celsius or 145 degrees Fahrenheit for 30 minutes. You have the flash method. That's where you go to 72 degrees Celsius or 162 degrees Fahrenheit for 15 seconds. And you have what's called the ultra high temperature method. You go to 134 degrees Celsius or 273 Fahrenheit for 1 second. Ultra high temperature sterilization is going to involve flash heating to rid the liquids of all living microbes. That involves processing liquids through a superheated steam and cooling it very quickly. So you get those temperatures up to 140 degrees Celsius or 280 degrees Fahrenheit, and you keep it there for 1 to 3 seconds before cooling. The next type we're going to talk about is going to be dry heat. So with the dry heat, that's going to be used for substances that cannot be sterilized by moist heat, such as powders and oils. It will denature the proteins and oxidize the metabolic and structural chemicals. It requires higher temperatures for longer periods of time, and so that dry is going to penetrate more slowly. Typically, it's going to be used at temperatures and times like 171 degrees Celsius for 1 hour, or that's 340 degrees Fahrenheit. Or it could go to 160 degrees Celsius, which is 320 degrees Fahrenheit, and you would do that one for 2 hours. So those are your 2 temperatures. This, you can also have a complete incineration using dry heat. That means it is going to be an ultimate means of sterilization. It's used to sterilize materials such as wire loops and glass spreading rods. The temperature ranges are going to be around 1500 degrees Celsius. That's 22,732 degrees Fahrenheit. Next, we have refrigeration and freezing. This is going to inhibit the growth and reproduction of most microorganisms by stopping their metabolic process. So refrigeration is going to use temperatures between 0 degrees Celsius or 32 Fahrenheit and 7 degrees Celsius or 45 degrees Fahrenheit. If you're freezing it, those temperatures are going to be below 0 degrees Celsius or 32 degrees Fahrenheit. Slow freezing is going to be the most effective means of sterilization, or stopping the growth. This is going to cause ice crystals to form, which are going to puncture those cell membranes. And so that's going to keep that cell from reproducing and growing. We also have what we call desiccation. That's a process of drying the organism out. It's going to inhibit growth as a result of the removal of water. And we have lyophilization. I don't know why I was having problems getting ready to say that. So with the lyophilization, that process is going to be basically freeze drying. That is going to preserve the technique in which microbes and cells are going to be frozen in a culture of liquid. It could be in with liquid nitrogen or frozen carbon dioxide. This is going to prevent the formation of large ice crystals. We have a filtration that we can use. Now with the filtration, that's going to be a passage of air or liquid through a material that's going to trap and remove the microbes. This is going to be used to sterilize things like liquids and gases. So what happens is the organisms are going to be removed and eventually killed by heat exposure or other treatments. Two different types of filtration methods can be used. You can use what's called a membrane filter, and that's going to be used for liquids. That's going to consist of a thin disc containing small pores, and it's going to take and trap those small viruses and large protein molecules. You can also have what's called a high efficiency particulate air or HEPA filter, and that's going to be used for air, and it's going to trap microbes and small particles in a filter. And then you take that filter, you change it out, and you throw it away. The last one we have on here is going to be your osmotic pressure. Now we know with osmosis that deals with the diffusion of water. So here we have the pressure that's going to be exerted across a membrane by solutes in a solution of one side of the membrane to the other. This is going to involve the use of high concentrations of things like salts or sugars. And so it's going to create a hypertonic solution. That is going to inhibit microbial growth and cellular metabolism as a result of the removal of water from the cell. So basically it kind of dries that cell out. Now we're going to talk about radiation. We have ionizing radiation and non-ionizing. So with the ionizing radiation, that's going to use wavelengths that are shorter than 1 nanometer. So that's going to include things like X-rays and gamma rays. These are going to create ions that produce effects that lead to denaturing of important molecules and then cell death. So it's going to be used to sterilize heat-sensitive materials, things like plastics, vaccines, and drugs. This is a very effective method, but it's going to be used less commonly as a result of its expense, so it can be kind of expensive, and it has a potential to be harmful to those that are applying it. With the non-ionizing radiation, those wavelengths are going to be greater than 1 nanometer. That's going to include things like ultraviolet light, visible light, infrared light, and radio waves. So with UV light, that's going to be the only type with sufficient energy to be practical antimicrobial agents. This is going to cause formation of what's called a pyrimidine dimers, and that results in mutations and death. So that helps to inhibit replication of these microbes. It's going to infect structures of proteins and nucleic acids, and it's going to be used to disinfect things like air, transparent fluids, and object surfaces. So you can actually take a UV light and disinfect things with it. Another fun fact, you can stick things like rugs and carpets, blankets, towels out in the sun, and that UV light coming from the sun can help to disinfect those things. Now, you might have heard biosafety levels or biohazard levels. There's going to be 4 levels of safety in microbiological laboratories. So if it's a BSL 1 or biosafety level 1, the handling of those pathogens do not cause disease in healthy humans. So that would be a basic average lab. In a BSL 2 or biosafety level 2, handling of moderately hazardous agents will be common. If it's a BSL 3, Handling of microbes is going to be in safety cabinets. And then BSL-4s, handling of microbes that cause severe or fatal disease. So that's your highest level of lab there. So if it's a BSL-4, it's going to have a lot of safety regulations because those microorganisms there are deadly. Now going into the different chemical methods of microbial control, these are going to be used for things like disinfection, antiseptics, and preservation. So these are going to affect microbial cell walls. It could affect their cytoplasmic membranes. It could affect the proteins that are being made there and also, or the DNA that's being made. So these effects are going to vary with differing environmental conditions. So it could be things like temperature, length of exposure, the amount of contaminating organic matter. It could be pH concentrations, or it could be chemical freshness even. Most effective against enveloped viruses and vegetative cells of bacteria, fungi, and protozoa. So that's what it's going to work best with. There's going to be 9 major categories of these. Looking at these first ones here, we have the phenols and the phenolics. So with the phenolics, those are going to be derivatives of phenols with the addition of a halogen or organic functional group. The level of activity here is going to be low to intermediate. And the mode of action with these is it's going to denature proteins, so it breaks them down. It's going to disrupt cell membranes, and then it's going to kill the cells. Alcohols. The level of activity here is going to be intermediate, and their mode of action is to denature proteins, disrupt cell membranes, and kill cells. You also have halogens. Halogens are going to be things like iodine, chlorine, bromine, fluorine. Their level of activity is also going to be intermediate, and their mode of action is to denature proteins, so break those proteins down. Oxidizing agents, things like hydrogen peroxide, are going to have a high level of activity. Their mode of action is to denature proteins through oxidation. You also have surfactants. These are going to be things like soaps and detergents. Their level of activity is going to be low, and their mode of action is to disrupt the cell membrane and cause lysis. Detergents and soaps are going to be emulsifiers, and the cell membrane is made up of phospholipids, so that's fats. So think about Dawn dish detergent. It goes in and it can break up that cell membrane, and that's what these surfactants are going to be able to do. Heavy metals are going to be things like arsenic, zinc, mercury, silver, copper, and lead. Their level of activity is going to be low, and their mode of action is going to be to denature proteins. Aldehydes are going to be things like formaldehyde. Their level of activity is high, and their mode of action is to denature proteins. Gaseous agents, things like ethyl oxide and propylene oxide, they have a level of activity that's going to be high, and their mode of action is to denature proteins. And then you have enzymes. Their level of activity is also going to be high, and their mode of action will also be to denature proteins. So antimicrobials are going to include 3 different types of compounds. You have what's known as antibiotics. These are going to be agents that originally were produced by some other living organism. Most are originally produced by either bacteria or fungi. You have semisynthetics. These are going to be chemically modified antibiotics. And then you have the synthetics. The synthetics are going to be wholly synthetic antimicrobial drugs. With all 3 of these, they're going to be used to treat disease. Their level of activity is going to be low to intermediate, and their mode of action is going to act on metabolism of those cells. It could act on the cell walls or the cell membranes. It could act on the protein synthesis and disrupt it, and DNA transcription and replication, so it can mess up that process there. Unfortunately, we have the development of resistant microbes with these forms of action. There's little evidence of extensive use of antimicrobials enhancing human health. It does promote development of microbial strains that can become resistant to antimicrobial chemicals, and that means susceptible cells die, and the ones that are resistant are able to grow and continue to proliferate and divide. So that's why we try to limit the use of food handling in healthcare with these types of antimicrobials, because we don't want to increase the ones that are going to be what we call antibiotic resistant. So with these antimicrobial drugs, the drugs are going to be used to, like we said, to treat those different types of infections. And the history of these are going to come from these men that we see here. So we have Paul Ehrlich. He's going to coin the term the magic bullet. And that basically is a chemical that could kill microorganisms but remain non-toxic to humans. He developed the first effective cures for certain diseases. He has salvarsan, that's an arsenic compound that's going to be effective against syphilis. Atoxyl, that's a chemical compound that's effective in treating sleeping sickness. And these are going to work, and discoveries are going to start to begin in the field of chemotherapy from his research. Next, we have Alexander Fleming. He's going to develop the earliest form of penicillin. It was actually done by accident. He was growing bacteria on petri dishes, and he realized that his dishes had become contaminated. And before throwing them out, he looked at them closer and noticed that around some of the microbial growth, where there was contamination, that it had cleared the bacteria that he was growing on those. And so that led him to wonder, what is this that contaminated my plate, and how did it kill the bacteria that's around it? And that's how we came up with our first antibiotics, penicillin. Next, we have Gerhard Domek. He's going to be responsible for developing the antimicrobial agents sulfanilamide. This is going to help to treat a wide variety of bacterial infections. So it's going to be an antibiotic that belongs to the class of drugs known as sulfonamides. These are going to treat things like urinary tract infections, strep throat, and ear infections. And what they're going to do is they're going to work by inhibiting the synthesis of folic acid. And folic acid is going to be very important to the bacteria that are susceptible to these, because that's what they're going to use in order for their nutrients for growth. And so that's what he discovered. Then you're going to have Selman Waksman. He's going to discover microbial agents produced naturally by microorganisms. And then he's also going to coin the term here, antibiotics. Before that, we did not use the term antibiotics. Right, so going in, we have the inhibition of cell wall synthesis, and that's going to be very important here. So first we need to talk about something called selective toxicity. That's going to be an effective antimicrobial that's going to be more toxic to the pathogen than to the pathogen's host. So we need to find something that is selectively toxic. Mechanisms for this, you can have several different groups of drugs based upon action. You have some that are going to cause the inhibition of cell wall synthesis. And so the inhibition of these bacterial cell wall synthesis is going to go in and inhibit the cell wall synthesis by interfering with peptidoglycan cross-linking. It also is going to cause cell walls to be less resistant to osmotic pressure, and they're going to lyse, or they're going to burst. It only prevents bacteria from increasing cell wall material, and it's basically going to have no effect on pre-existing peptidoglycans. You also have inhibition of fungal wall synthesis. That's going to inhibit enzymes that are responsible for synthesizing glucan. Glucan is going to be a component needed to make cell walls, and that's going to cause fungal cells to lyse due to less resistance to osmotic pressure. So it allows too much water in, and it's going to cause those to burst as well. So remember, lysing is bursting. Second up here, we have the inhibition of protein synthesis. That's going to go in and inhibit the initiation of protein synthesis or making proteins. It inhibits codon recognition and the docking of the tRNA amino acid complexes at the A site of the ribosome. Disruption of cytoplasmic membranes is going to be a third way. This is going to disrupt the membrane by creating a channel through the membrane, and that's going to damage or mess up the integrity of that membrane. How it's going to do this is it's going to attach to and affect the production of ergosterol that's in fungal cells, and that's going to cause that cell to lyse, or the ability of that membrane to remain intact, and that's going to cause that cell to die. It's also going to affect the transport and permeability across cytoplasmic membranes altogether. You've got your fourth type up here that's going to be inhibition of metabolic pathways, and this is going to work by interfering with electron transport, enzymatic activity, and disrupt tubulin and polymerization. And basically that is going to be the formation of the flagella, and it can interrupt glucose uptake, so taking up nutrition and food. It can act as a structural analog for compound that's going to be needed in DNA and RNA synthesis, and it can prevent viral uncoating, so taking the coat off the virus, which protects it. The fifth way, we have inhibition of nucleic acid synthesis. This is going to block DNA replication and transcription and RNA transcription. It's going to act as a nucleoside analog. That basically is going to incorporate into DNA or RNA, distorting nucleic acid molecules and preventing further replication, transcription, or translation. What that's going to do is inhibit enzymes that are responsible for coiling and uncoiling the DNA, and it can inhibit the action of the RNA polymerase. And then the last one we have up here, the sixth one, is going to be prevention of virus attachment. So the attachment antagonist is going to be a structure that acts as either the attachment or receptor protein. And what it's going to do is it's going to prevent attachment and entrance of a virus to a host cell. And so it can't get in and it can't infect the cell. Now here we're going to look at a picture. We're going to look at inhibition of cell wall synthesis in things like penicillin and cephalosporins, things like that. So you see how it can't get in. You see inhibition of protein synthesis, so we can't make those proteins. We have disruption of those cytoplasmic membranes there. Going around from clockwise, we're going to have inhibition of general metabolic pathways, so it can't break things down. We have inhibition of DNA and RNA synthesis, so you see those. And then you have your inhibition of the pathogen actually being able to get into the cell. Now, here we're going to have a spectrum of action. What's going to be important is we have some characteristics of ideal antimicrobial agents. So they need to be readily available. They need to be inexpensive, so fairly cheap. They need to be chemically stable, easily administered, non-toxic, and non-allergenic. And we need it to be selectively toxic against a wide range of pathogens. So that's where we get the spectrum of action. We have a number of different kinds of pathogens that a drug acts against. So you have two different types of spectrum drugs. You have what's called a narrow spectrum drug. That drug's effectively going to control only a few or sometimes only one type of pathogen. So it's only going to affect and cause death for that one type. Broad spectrum drugs are going to be drugs that are going to be effectively in controlling many different types of pathogens. So it might be one that could affect many different types, let's say, of bacterial infections that you're going in and looking at. So here's one of our visuals that we can look at to see which ones are going to be most effective. Notice how erythromycin and tetracycline and the sulfonamides are going to be the ones that are actually going to be more of a broad spectrum coming in. We have polymyxin. We have, let's see, some of the smaller ones that are going to come in, and they're only going to affect small range of different types of bacteria or microorganisms. Now, in order for a drug to be used with the population, it has to pass efficacy tests. Efficacy tests are going to be used to determine the effectiveness of an antimicrobial agent in treating infectious disease. There's going to be three types of efficacy tests that you can use. We've got the diffusion susceptibility test. That one's going to be simple, it's inexpensive, it is very widely used, and has some basic steps. So basically, you go in and you inoculate a petri dish with a pathogen. Then you're going to place a small paper disc with drugs onto the plate surface. And so you see those discs there, and they're labeled with their drugs. Then you're going to let that plate incubate. The plate is then going to be examined for what's known as a zone of inhibition. Zone of inhibitions are going to be a clearing or a clear area where that bacteria did not grow. Larger zones of inhibition are going to be showing that those drugs are going to be more effective against that pathogen. So if it's a small zone of inhibition, it's not going to be quite as effective